So the next honor that I have as president of the society is actually selecting the guest of honor. And so, as I mentioned, the concept of bench to bedside, I think, is very important. And so this individual highlights that. And so I'd like to introduce Professor Marcelo Rivolta. He got his medical and surgical training at the University of Cordoba in Argentina, did his fellowship and doctorate work at the NIDCD at NIH, and then from NIH he held positions, but then decided to go to the UK, where he held positions in the University of Bristol and Sheffield. He has over 30 years of experience in the hearing field and in the use of stem cells for regenerative therapies. His lab identified and isolated a population of stem cells from human fetal cochlea and also generated one of the first developmentally informed protocols to direct human pleopotent stem cells into the auditory cell types. He's demonstrated this in preclinical models, the proof of concept that stem cells can be used to functionally repair the damaged cochlea. He's chair of the sensory stem cell biology at the University of Sheffield and the founder, director, and chief scientific officer of RenRe Therapeutics, and that's a biotech company that's advancing the clinical application for pleopotent stem cells for the treatment of hearing loss. He's working to develop clinical trials, which is actually hopefully going to be starting this year, and so he's going to tell you about that. So let me ask Dr. Rivolta, to come to the podium as our guest of honor. So please welcome him.
Right. Good afternoon, everyone. I'd like to start by thanking Bill and the committee for this incredible honor of inviting me and giving me this opportunity of telling this little bit of the history of a journey from the bench, hopefully very soon, to the bedside. So our story kind of started, before we do that, a disclosure statement. As we say in the introduction, I am the founder, director, and chief scientific officer of RenRe Therapeutics alongside my academic appointment. So we started this journey more than 20 years ago now. And in our lab, we wanted to develop what we saw an opportunity to bring a new kind of therapy to the field, which is the use of stem cells in a regenerative approach. And we think we had a very good case, and for the cochlea is a very good target for this kind of therapy because, as you are all aware, there is it's a huge health issue. There is no biological curative treatment. We have a very useful prosthetic devices, but so far we have nothing that can repair biologically the cochlea. And as mammals in general and humans in particular, we lost through evolution this ability to restore hair cells or other sensory cells in the system like the neurons. And because divisions only happen in utero. So we are born with a complement of cells which is meant to last a lifetime, and there is no really regenerative, proliferative capacity left in this organ. So we were worried that all these other strategies which are trying to awaken and put cells back into cell cycle will find really a very difficult, very challenging biological hurdle to overcome. However, if we try to make the right cells ex vivo and then transplanting them, that will be probably a more feasible approach. Also, the cells that, the cellular problem is the bottleneck, the common denominator of a lot of pathologies. At the end of the day, what we lose in the cochlea are cells. So that's also part of the justification that we can make these cells in vitro then hopefully we can use them to repair and replace the damaged organ. Logistically, we have also an advantage in this, in our system, that is we don't need that many cells. The normal complement of hair cells and neurons in a cochlea is about 16,000 hair cells, about 40,000 neurons in a human or mammalian cochlea. So from a point of view of manufacturing this is not such a big challenge. We have other challenges, which you will see in a minute, that we need to overcome. But it's not like, for example, if you're trying to regenerate a whole heart or a liver when you need 10 to the 7 cells or more. So conceptually, if we could make these cells ex vivo and then transplanting them, we thought, well, potentially we have a good case. I'm talking about this unmet medical need. Obviously, you all know that the hair cells are traditionally being considered the main cell type that when it's lost is responsible for the onset of deafness. But in the last decade or more, it has become very apparent that the neuron, the spiral ganglion neurons, is an incredibly important, and I would say sometimes equally or even more important cell than the hair cell. In many conditions, the neural damage precedes chronologically, but also in magnitude to the damage of the hair cell. So you will see in a minute that a lot of our efforts have been concentrated in restoring the neural health in the cochlea. We're also working with the hair cell, but that is at a much earlier stage of development. We are concentrating on developing a treatment for neural replacement. Also, because as you well know, for hair cell replacement, you can use this fantastic prosthesis, which is the cochlear implant, but if you don't have good nerve provision, then you will have a very limited performance of even a cochlear implant. And for the loss of the spiral ganglion neurons, we have basically no treatment. We can use sort of a brainstem implant, but as you well know, it has limitation and is not as well developed as the cochlear implant is. So we did a lot of early work in our lab because when we started this, we realized that we knew a lot about progenitor cells and stem cells in the mammalian system, particularly in the model organisms like the mouse, but we didn't know that much about how human cells really work and how we can handle them, how can we culture them, how can we treat them in the test tube in the lab. So we did initially a lot of work with cells, isolating cells from the human cochlea and adapting the methods that others and us have previously been used in the mouse system. And then we learned to identify this population of cells from human fetuses that then we learned to push them to differentiate into hair cells, but more importantly, into spiral ganglion neurons. But those cells have a very limited usage because you always have to go back to primary tissue, to this sort of tissues that came out from terminations. So we needed to look for a permanent supply of cells that you can produce the cells that you want on demand. And for that is where it comes this type of cells, which is called the pluripotent cells, which are there are two types of pluripotent stem cells. One is what is called the embryonic stem cells, and these are the first ones in the field that were identified initially by Jamie Thompson in 1998 in the University of Wisconsin. And it's cell lines that you can produce from inner cell mass of blastocysts and expand them in vitro, and then these cells have the capacity to produce any cell in the human body. What you need to learn is to decipher the cues that you need to give these cells, the chemical instructions to put them in the right developmental pathway. So embryonic stem cells derive from the blastocysts, established as a cell line, and then you can push them to make the differentiate into the lineage that you want. In our case, otic cells. The other cell type is a cell type which is artificially generated. It's called induced pluripotent stem cells, and this was a technology developed primarily by Shinya Yamanaka in Japan, and it's a process when you take a somatic cell, like a blood cell or a skin cell, and you reprogram these cells. You expose them to a series of transcription factors, and you turn the somatic cell into a pluripotent cell, just like the embryonic cell. So you have two different sources. In many ways are equivalent, the embryonic cells and the induced pluripotent stem cells, and we are working with both. To sort of get a little bit ahead of the presentation, most of the work that we have done and the initial clinical trials will be done with embryonic stem cells, but the work with the induced pluripotent stem cells is just shortly behind in the pipeline. The reason why we are pushing forward the embryonic first is because from the point of view of the regulators, it's much more acceptable because these cells haven't had any kind of genetic modification, while the other ones have been reprogrammed and reset. But these days, and there are quite a few clinical trials already in other fields taking place using embryonic stem cells. But both of them should be considered to a point equivalent from the point of view of the type of cells they can produce. So what we did more than 10 years ago now is to develop a method that will take these pluripotent cells into becoming ER cells. So it was a developmentally informed protocol. So what we're trying to do is generate in the tissue culture the same kind of stages that you will see happening in vivo during development. So we push these cells through a series of chemical instructions to become otic progenitors. Then we incorporate the purification step, and then we can isolate and create these otic neural progenitors that can be expanded further and then used to differentiate into neurons in vitro or put in vivo in an animal model or eventually into a human patient. The protocol is relatively simple in the sense that it's conceptually simple, and it has modular stages. So you can take them to different parts and stop them and perhaps free cells at different points, making the application of this more amenable and more useful. So after that initial demonstration in vitro, what we did then is testing if these cells that we were manufacturing were competent in vivo in an animal model. So for that, we used the gerbil. As you know, the gerbil is an excellent model of hearing research, mainly because all the frequency range in the gerbil is very similar to the human frequency range, and also because there is a model of neuropathy which was developed in the gerbil by applying wabane, a neurotoxin, directly in the rung window of these animals. It's a model that was developed by Schmidt and Lang in South Carolina and that we have refined in Sheffield, and it produced a very clean ablation of the type 1 neurons, spiral ganglion neurons in the cochlea. And in that context, we delivered ourselves in the modiolus of the cochlea of the gerbils, and we saw restoration of the auditory brain stem thresholds and engraftment and differentiation of the cells. And we were able to monitor that initially for about 10, 12 weeks. So after that initial proof of concept, we published that in Nature, as I said, about 15 years ago now. We have improved the protocol and make modifications to the protocol. One of the things that we did is concentrating in that purification stage, because although the protocol is very efficient, you always produce cells which are off target, cells that you don't want. And we want to have a very clean population of otic progenitors, which is going to be a therapeutic agent, a therapeutic product. So for that, what we use is a technique which many of you may be familiar with, but certainly it's very common in hematology, which is a technique that you use to purify cells, and it's called a fluorescent automated cell sorting. For that, you need to have a cell line that is labeled with a particular fluorescent tag. It could be a reporter gene, or it could be an antibody that is coupled to a fluorochrome, and that will identify a subpopulation of those cells. And then you put it through this machine that produces little droplets containing single cells. It's activated with the laser, and that separates, the positive cells, and then you put it through a nano-nanocathode and separate the positive and the negative population. And then you get two peaks of positive and negative cells. And that way, you can separate cells based on this fluorescent marker. So what we did was generating a line that had a genetic reporter, green fluorescent protein, that it was controlled by a genetic sequence which it will only express in the nasal and the otic placodes, and that was basically producing green fluorescent otic neural progenitors. And this is the plot before the sorting, and this is purified after the sorting, and we get almost 99% purity of our cells of interest. So these cells then, we put them back into our gerbil model, and we monitor that for a long period of time. We followed these animals for up to a year, and at the point of termination, we did a whole body scan using MRI scanning of the head and the whole body, trying to identify potential lesions, potential areas of tumor formation. Because one of the concerns, historical concerns, and so far I would say unfounded concerns on stem cell therapy is the possibility that these cells will produce tumors. So we want to check that with the cells that are safe. So we monitored these animals for up to a year, and we did the scan. Any potential area of concern, it was explored further in a full post-mortem, and we found no evidence of formation of tumors, anything that could be attributed to the test article, to the cells that we were testing. We also did biodistribution analysis, that is basically taking samples from different parts of the body and looking if the cells have migrated out of the cochlea when they were delivered. And this is an analysis based on PCR, looking for human-specific ALU sequences that will obviously be distinguishable from the genome of the gerbil. And we saw that no cells, all the results, all the tissues explored were below the limit of the detection of the technique. There were no cells escaping from the cochlea. And there was a long-term maintenance of the recovery. It was tailing off after 37 weeks or so, because there are other comorbidities that the animals, which are all start to develop, but it's still performing much better than the control that have received the same treatment, but only the vehicle, not the cells. We also wanted to explore whether these cells, because remember here we're trying to replace the spiral ganglion neurons, we wanted to see if these cells could interact with the cochlear implant. So for that, we used a prototype of a cochlear implant in a gerbil. This was an electrode that it was sort of facilitated to us by Cochlear, and is, because of the small size of the animal, the electrode is a fully implantable electrode and has this chip in the back of the animal. The electrical stimulation goes to this part, which is inserted inside the cochlea like a normal cochlear implant, but instead of being acoustically stimulated, is stimulated by a magnetic field, by a pulsating magnetic field. So the chip in the back of the animal is on this kind of mini chamber here that creates a magnetic field that delivers electrical pulses, and in that way you can measure electrically induced ABRs. And what we saw in this model, these animals had been going through the Wabane ablation protocol, so they have all the neurons depleted and also have lost the hair cells because they were treated with aminoglucosides. And in this background, what we saw was, again, engraftment and integration of the cells, formation of fibers that will go and contact the electrode, and then restoration of the electrically induced ABR. This is the kanamycin furosemide-treated animals that have the endogenous neurons, so this was the normal neural responses that were detected, and when they were transplanted with the cells, we get a restoration of the similar profile at about 5-6 weeks post-transplant. The animals that received only the vehicle, they didn't have any restoration of the electrically induced ABR, only the stimulus artifact that you get at the beginning of the process. We then went back to revisit the purification strategy, because I was saying before we were using the genetically modifying lines, but those lines, because of the genetic modification, will not be suitable for clinical applications. So what you're trying to do is trying to modify or introduce the minimal kind of changes to the cells. So instead of using a genetic modification, what we did was identify cell surface markers that we can use to purify the cells using an antibody, and that's what we did. We managed to get very highly pure population of cells that were resembling the other ones that had been purified genetically, and showing that they can be efficacious in the animal models as well. Then, if we move to later in the process of making the cells, we are here in the phase of expansion. What we wanted to do is to characterize this population of cells through the expansion phase. You want to make sure that you know and you understand your cellular material, you know what these cells are, and also that your process is reproducible. So here you have two different batches, independently produced batches of cells, batch 1 and batch 2, and this is data from single-cell RNA analysis. So each point here represents a single cell with the whole transcriptome has been analyzed, and you have then aggregated in different clusters which represent slightly different stages in the development and differentiation of these cells. But as you can see, the profile of this batch is almost identical to the profile of this batch, and then 2 passages later, you have a profile which is also completely reproducible by a different batch. So this is the stages that we are taking ourselves, that we think ourselves are suitable enough then to be transplanted. So with all that information, we wanted to then move forward and closer to a clinical trial, and then you get what it has been described as the valley of death, because moving something from the bench to the bedside, to a real patient, it takes a lot more work and a lot more challenges, and also funding that sometimes is very limited and is very, very kind of like, it's very short-sighted. You can only go one step and then who knows. So in order to facilitate this challenge, I decided at that point that perhaps the best way of doing it, it's not the only way, but perhaps the best way of doing it was setting up a company, and then founded in 2018, Reentry Therapeutics, with the support of Biosciences, UCV Ventures, Boehringer Ingelheim, and the University of Sheffield. So since then, we have sort of kept moving into this journey to the clinic. The preclinical development that you need to do is a quite complex journey, as many of you will know. You need to go through making your process, your manufacturing process, compatible with moving it away from the academic setting and compatible with something which will be acceptable from the point of view of a clinical standard. You need also to fulfill and be aware of the regulatory limitations and requirements that you need to fulfill during the pathway. There are elements of safety that we need to develop even further from what we have already done, and also elements of clinical development that we needed to cover. So basically we had these 4 pillars of process development, preclinical safety and toxicity, regulatory and clinical standards that we needed to expand. So the next few minutes, I will try to briefly touch on what we did there. So this is, again, that schematics of the manufacturing process, something that we have created in the lab, of how to make these cells from a pluripotent stem cell population into becoming these otic neuroprogenitors that can be used in vivo. But there are different steps in the process that need to be adapted away from the academic method into a proper industrial application. So, for example, the initial part, when you're handling these pluripotent cells, they are grown on what is called feeders, which initially were mouse feeders, then were human feeders. Feeders is basically a layer of fibroblast cells, which is very useful from research, but makes the manufacturing of these cells very complex and, in many cases, unacceptable. So what we needed to change that and identify a matrix that will be recombinant, a matrix that will be produced in a different way, and it will be acceptable for good manufacturing practices, which is the standard that you use in a clinical setting. Then the differentiation protocol had also components which were from animal origin, for example, BSA, laminin, that needed to be adapted and replaced for cell therapy grade or GMP grade reagents, and also trying to move away from any kind of animal component that may have been in the original process. The purification step I was mentioning before, we move away from the use of a genetic reporter or an open sorting into surface markers and a closed sorting, when you separate the cells in a chamber that is not exposed to the air at all, so there's no possibility of any kind of contamination. And all these steps that they sound very trivial and simple, they demand a lot of testing and characterization. And then the expansion phase, again, you needed to remove a lot of the animal components from it. So then, that's why we sort of arrived to this process that we have now, which is CMC, sort of chemical manufacturing and control compatible. And we go all the way from the starting material, which is our pluripotent cells, and we have banks of the pluripotent cell line, which have been manufactured in a cell manufacturing organization, all the way to the product that is cryopreserved that we can use, hopefully, in a trial. At the moment, we have transferred our technology to these manufacturing establishments, and we have done 2 runs now of what is called engineering rounds. This is the rounds when you test that everything is working well, and the final run we have tested it in the animal models and show that it's efficacious, so we can completely produce a clinical material or a material of clinical grade that we know is efficacious in the animal model. And we can scale it up and hopefully cover the demands that this will have. For the point of view of the preclinical studies, we did a lot of the safety studies in the gerbil model, but from the point of view of toxicology, the gerbil is not a conventional model. It's not a model that is well known. So we had to move to the rat. The rat is a very well-established model for toxicology. And for that, we use this rat, which is called the STRG rat. It's a rat that is an immunosuppressed animal. It has a series of mutations that it makes it completely immunosuppressed, because obviously being a xenotransplant here, we need to provide immunosuppression to the gerbils. But in this way, using this genetically modified rat, we can test for safety without the potential confounding effect of the cyclosporine that we were administering, but also because it's so severely immunosuppressed, any potential concern, anything that the cells could be doing wrong, will be easily spotted in this model. And we did the studies in the rat. We show very nice engraftment and integration. You see fibers of the transplanted cells there contacting the hair cells, engrafting very well in the rat model. And we did 2 studies under GLP, so this is the kind of regulatory standards that you have to use in the lab to fulfill the regulators, and show no concerns, no evidence of any kind of toxicity attributed to our cells in this model. The regulatory pathway, you have to interact with the agencies. You very well in the US is the FDA, but in the UK is the MHRA. And we have just about 2 months ago, in February, we submitted our clinical trial application to the MHRA, and we're just waiting for the final approval. But one of the things that we did from very early stage is work closely with them and getting their advice and making sure that everything that we were proposing to do, like for example, the testing strategy that we had developed, they were happy with that and having their endorsement. And hopefully everything is fulfilling these kind of regulatory steps. And finally, the kind of development that we've done clinically to take this all the way. One of the things that we needed to adapt is the way that we deliver the cells, because in the way that we do it in the gerbil, we go through the round window and we drill into the nerve canal on the gerbil and the modulus. And this is a CT scan in the gerbil here, showing when this is the cochlea there, and this is the base of the cochlea, and this is where we deliver our cells. This is pseudocolor here in blue. But that approach doesn't exist for humans just yet. So in collaboration with different groups, particularly with the laboratory of Professor Helge Raskanderson in Uppsala, and the groups in the Western Ontario, Western University in Canada, and Guy St. Thomas and the University of Nottingham, Professor Doug Harley, which is a very close collaborator, colleague, and member of our chief medical officer in RINRI now, using synchrotron images, high-resolution synchrotron images of the cochlea, the group in Uppsala, describe a safe passage to the base of the cochlea when you can reach the nerve compartment here, the inner, the internal auditory canal, the internal auditory meatus there, and through the round window without sort of damaging any of the key elements, anatomical elements on the base of the cochlea. And the surgeons reproduced that, tested that in several cadaveric bones, and delivered the suspension of cells in these temporal bones that they were able then to reconstruct with CT scan, and showing that we have now a route of administration that resembles and reproduces the one that we have used in the animal models. And we have done several surgical simulations in theater now, testing, and it's one of the sites that we're hoping to become active soon, testing all the cellular pathways, so how to move the cells from the point of view of origin all the way into the theater, just making sure that we have all that route properly mapped out. Then the strategy for the clinical trial will be, in its initial first in human trial, will be in combination with a cochlear implant, will be as a sort of associated as an adjunct therapy with the cochlear implant. There will be patients that will have the cochlear implant on its own. This is the standard of care. And then patients that will have our cells together with the cochlear implant. One of the things that Doug Hartley has invested a lot of time doing as well is making sure that we can produce some efficacy recordings of potentially the material after it's been transplanted, and for that, we are going to be leveraging the cochlear implant itself. So we're going to be using primarily the advanced bionic implants. We can use the tablet for the telemetry, and they can you can sort of use and read the compound action potentials from the electrodes. And Doug has done these preliminary studies showing in acutely transplanted, but also in chronically transplanted patients, you can visualize areas, for example, here, electrode dead regions, which you have completely unresponsive areas which remain unresponsive for the duration of this study, which was about 3 months. So hopefully these are the areas that we know once you detect them in the early phase, hopefully then we could try to revert them with the application of our treatment. And because we know, based on the animal study, that there is a lag phase between the moment that you transplant the cells and the moment that you see kind of starting to see activity in the animal models, that takes about 4 to 5 weeks, you have time to make that baseline recording of your kind of like your dead region, and then hopefully seeing activity coming up in those areas. Again, also developing imaging protocols to look for the nerve space and making sure that the cochlear implant will not interfere with the visualization of these areas to be able to detect any kind of concern, anything that may be sort of developing in those areas. But again, based on the work that we've done, the extensive work that we've done in the preclinical models, there is no indication that these cells will do anything unwanted in that regard. So the patient population that we're going to be targeting is going to be two groups. One is going to be presbyacusis, sort of age-related hearing loss. As I said at the very beginning of the talk, there is an increased body of evidence produced by different labs, but primarily by the group of Charles Lieberman, showing that there is a loss of innervation and a loss of neurons that that occurs with age. And this is, for example, here, data from one of their papers when you show the loss of spiral ganglion neurons. In gray here, that is in age-related hearing loss, that from mid-frequency, sort of over 2 kilohertz, 40 to 60% of the neurons are lost after 60 years of age. So these patients are going to be patients that are eligible for cochlear implantation in the UK, over 60 years old, and they are going to be implanted, and going to be the dead regions are going to be mapped, and then the monitor for the progress of the regions to see how they evolve. The other patient groups is going to be the conventional postsynaptic auditory neuropathy. These are patients which are usually younger than 60 years of age, but again, eligible in the UK for cochlear implantation, but they have these kind of pure neuropathies. They have presence of autoacoustic emissions, representing that they have preservation of at least outer hair cells, but the ABRs are absent or extremely abnormal. And although these patients are eligible for cochlear implant, normally they don't perform very well with the cochlear implant. So this will be the other patient groups. And both of them, they're going to be these two cohorts. They're going to be about 6 individuals that are going to receive the treatment and the cochlear implant, and 4 that will be the standard of care, just the cochlear implant. So in total, about 20 patients. Going to happen in 3 different sites that we're hoping to be able to open sort of late in summer, beginning of the fall. And obviously, the primary readouts are going to be safety. This is a phase 1/2A study, so the primary readouts are all related to safety, but also we're looking for early indication of efficacy and feasibility. The early indication of efficacy is going to be the caps using the electrode, but also they're going to be a sort of speech perception and the more sort of conventional tests, which are going to be obviously a higher impact if there is an improvement of those metrics. So to summarize and to conclude, we have done a series of preclinical studies with the name of our cellular material. We call it RINCEL1. We've done a series of preclinical studies with RINCEL1 that confirm that it's safe, effective, and remains localized at the administration site. We have taken the academic process away from the academic university lab into a manufacturing organization and taken to meet the standards that are required for the regulatory production of cellular material. We have done a series of clinical developments to be able to deliver these cells, but also to make the kind of right informed outputs out of this material. And we are hoping to be able to start clinical trials, as I said, in the next few months in the UK. And finally, just closing and to thank the people involved in all this, here showing my academic lab together with the RINRI lab. A lot of the work that I presented today was done by Daniela, which is also attending at the conference today. And I'd like also to primarily thank our clinical team led by Doug Hartley, which is our Chief Medical Officer, but also Rachel Hines, which is our Vice President of Clinical Operations, and Cherith Campbell-Bell as Director of Clinical Development. Well, thank you very much. Thanks again for the honor of this invitation, and I'm happy to take questions.
That was an exciting talk, so we have time for a few questions. Simon?
Dr. Rivolta, that was wonderful. Thank you. You showed some beautiful slides of engrafted cells. Were those pluripotent cells or already differentiated auditory nerve fibers? And how long do they last, the transplanted one?
Yeah, no, thank you very much. Very, very good question. The pluripotent cells is the starting material. We never transplant pluripotent cells because that will be a potential risk. Actually, part of our strategy is to purify out any remaining pluripotent cell. So we push them to differentiate into becoming these otic neural progenitors, the ring cell 1 cells. That takes about 2 weeks into getting the right cells. Then they're purified, and the purified cells are expanded further. So those are the cells that we transplant, and they engraft. How long do they last? Well, in the animal model, we know that they can last at least or up to about 40 weeks. After that, functionally, you see a decay, but it's a decay because the animals are getting very, very old. If we translate that to a human population, that will be probably equivalent to decades. But it's one of the things that we need to sort of confirm in the clinical trial, how long the engraftment will last in a human. We think that because we are mismatching species, we are seeing here the worst case scenario. The humans, because they're going to be the same species, it's likely that's going to last even longer.
Good, Jay?
Great talk, thank you. Given the variability of cochlear implant outcomes, how many subjects do you think you're going to need to enroll in the trial?
Yeah, the first trial, we are only using about 20 patients, 16 actually implanted with the delivery drug, because we have the 2 dimensions, which is the longitudinal analysis, so we have the baseline, and also we have the multiple electrodes that will allow us to distinguish different zones. So we have powered that study to get an initial efficacy. It's not a very powerful study, we know that, but because the main objective of the initial trial is going to be safety. But with those numbers, we could measure a significantly different improvement if there is there.
Thank you.
There's somebody under the light. Just say your name and where you're from.
Hey, my name's Dan Romano, one of the residents at Washington University in St. Louis. I actually have 2 questions if we have time. My first one is regarding the preclinical model. It seems like these animals were acutely deafened with kanamycin and furosemide. I'm curious if you have any thoughts on whether the cellular host environment may be different in this sort of acute deafening versus the more chronic degeneration that occurs in presbycusis, and if that might affect the efficacy of this intervention.
Yes, again, a very good question. It's true, it is relatively acute, although we have done different experiments waiting for different periods of time, and we don't see much of the difference. The rat model is not an efficacy model, so there is no an acute intervention in that sense, and the engraftment seems to be fine, robust, and the cells seem to be doing the right thing. So we don't think that that is much of a concern.
Do I have time for one more question? Yes. My second question is regarding the use of ESC-derived otic neuronal progenitors. Do these patients need any sort of immunosuppression, given that they're allogeneic transplant versus like a iPSC, which would be more autologous?
Yes, but what we have to be aware is that autologous iPSC therapy is not here just yet. There's only been 1, potentially 2 trials at the moment using autologous iPSCs, because autologous iPSCs, the concept behind that is that you will take a somatic cell, produce an iPSC line, and then put it back into the same patient. But in order to do that, you face 2 big challenges. One is the cost of that. It has been estimated at the moment is about $3 million per patient to do that. Although that may be changing because Japan is developing a very high throughput technology that is bringing those costs down. But the second problem that you're dealing is that every time that you do that, you need to certify the making of these cells, which are absolutely perfect. There is no genetic modification, there's no genetic errors, and that has to happen every time that you make a line. The first trial that was done in Japan in 2017 using autologous iPSCs, they generated 5 lines for a patient, and they couldn't transplant. They decided not to transplant any because there were genetic modifications that they couldn't explain. Not necessarily they were negative, but they just couldn't interpret it. That's why ES or allogeneic, it is still having a big space, but also the use of iPSCs as allogeneic. So what you do is you create iPSC lines from a series of representative of the population, you characterize those lines really, really well, and then you use it in multiple patients. So allogeneic therapy, even with iPSCs, is going to be probably the way forward for the next, I would say, maybe 10 years. There is also a completely different arm which is being developed at the moment, which is making hypoimmune pluripotent cells. So you change the immune makeup, and in that way you make it like universal donors. And that will probably be the way forward rather than autologous, because autologous has still big problems and big limitations.
I'd like to, I think if you all would join me in giving applause to Dr. Rivolta. I think it's very ambitious for every scientist to say, I want to go from bench to bedside, but you've actually crossed the valley of death and you're getting into the clinic. So again, congratulations for your work. Thank you very much for sharing with us.
Thank you.